Co? Good afternoon. On behalf of the organizing committee of the 23rd International Medical Students Conference in Kraków, I have the great pleasure and honor to welcome you to the closing ceremony. I welcome our special guests, Professor Beata Tobiasz-Adamczyk, representative of the Rector of Jagiellonian University for Education and International Cooperation at the Medical College. Professor Tomasz Grodzicki, Dean of Faculty of Medicine. Professor Tomasz Guzik, Mentor of Student Scientific Society. Dr. Jerzy Frydyger, Vice President of the District Chamber of Medicine in Kraków. And Dr. Bartłomiej Guzik, Secretary of the District Chamber of Medicine in Kraków. This year, we received over 450 research papers from 36 countries. We are extremely happy that this event is growing in size and it's well recognized all over the world. The achievements of this conference would not be possible without the dedication and time of the organizing committee, coordinators of individual sessions and workshops. I would like to sincerely thank all of my colleagues for their very hard work in the past few months. We would like to emphasize that organizing the Students' Conference would not be possible without kindness and support of authorities and professors of the Jagiellonian University. We are also grateful to the District Chamber of Medicine in Kraków and all scientific societies for their patronages and contributions in organizing of this wonderful event. Lastly, I would like to thank the students' governments of our university and the local committee of International Federation of Medical Students Association in Kraków for their cooperation and support in planning this event. Also, I would like to thank our sponsor, without whom getting all the prizes would not have been possible. I would like to ask Professor Beata Tobiasz-Adamczyk for a few words.
Esteemed Dean and esteemed professors, dear colleagues, each conference has own cares. Usually, as this conference started with opening ceremony, this year we had opportunity to listen very nice concert given by young musicians, and the singer was students of medicine. Yesterday, we had the opportunity to participate in different sessions and also had the opportunity to hear very impressive lecture given by Professor King from Boston. And today is a closing ceremony, which usually is associated with some congratulations, awards, and thanks. At first, I would like to thank all of you that you are come here to Krakow and participate in this conference. Usually, the conference consists of not only medical students, but also students from pharmacy and health science. Besides of scientific program, you had also opportunity to create your social life ties and good social relationship, which will be continued in the future. I would like to say that I am very grateful for you because I appreciate very much your significant contribution in science, and it is very important because when I participate in several meetings associated with education and also medical education, I very often heard some opinion. From my point of view, is unfair opinion about the medical students. So of course you are the best students thinking about other students, but you are very much concentrated only on everyday curriculum. That you are thinking only about the medicine, about the lectures, seminars, and exercise. Nothing else. And you as a person, as a medical student, but also students for health science who are involved in the research activity, who build or develop the new ideas for medical research, today research and future research, you are able to copy with this unfair opinion. You are able to create the new image of medical students' situation and opinion in society, but also among people who are authorities for medical education and also general education. So for this reason, I am very grateful for you. And of course, I would like to congratulate all the winners of the sessions, but I would like also congratulate to all of you. And I was very happy that it is possible to also thinking about you as a person who made a great effort to the science, but also thinking about you as a students who maybe spent a nice time here in Krakow and have a good opportunity to also create some relaxed atmosphere among their friends. So thank you very much for coming here. I hope that in the next year will be possible for me to meet you again here in Krakow. And I would like to thank also the Katarzyna Konarska and the organizing team, because you know, conference means something, but behind the scene, it's a hard really work, and I know about it because I accompanied the organizing committee, and usually this work starts immediately after the ending ceremony of this conference. So congratulations, and hope to see you next year in our university. Thank you.
And I would like to ask Professor Tomasz Guzik for a short speech.
Thank you very much for this opportunity, and thank you for an opportunity to listen to your research, because I enjoy this every year, and I truly appreciate the fact that from year to year, the research presented by students, by you during this conference, becomes more and more advanced and uses more and more complicated and more modern methods and appropriate methods. Size of studies, size of study groups is increasing, and we can see that from year to year we see better research being presented. And the truly wonderful thing to participate in, for which I thank you particularly strongly, is your curiosity. It's curiosity that brings you here, that brings me here, and I wish you and myself that we do not lose this curiosity, and we will remember that being a scientist is actually a wonderful opportunity, because I have a friend who is a really excellent scientist in the United States, and he always says that we are very lucky because we are paid for doing our hobby. So let's do our hobby. And congratulations for this conference, because it is truly marvelous. Thank you.
And now I would like to invite you to the closing lecture of Dr. Paweł Wołkow, head of the Center of Medical Genomics Omicron, and entitled On the Road from Technological Breakthrough to Clinical Application in Genomics.
Thank you very much. I would like to start thanking the organizers of this conference and authorities of the university for the opportunity to tell you today a few words about genomics, which I believe is the very important frontier in biomedical sciences. I know I'm standing in front of your prizes, so I will try to be short, but I do need to take some time because I have a message that I would like to deliver. And okay, and I have a homework for you that I would like to think about before the next conference. So you have 1 year to complete this homework. My talk will be divided into 3 parts. I will touch upon technology, I will talk about applications, and then finally I will try to say a few words about the road ahead of us in genomics. When we talk about technology, I will be dealing with microarrays, with next generation sequencing, and with mass spectrometry for proteins. So as you can see, quite a lot to cover in just a few minutes. I don't want to bother you with technology too much. The point is that with microarray, you can amplify your DNA, you share it, and then you load it on specific chips. Chips can be used for various purposes. I will talk about that in a moment. The very important part of this reaction is hybridization of complementary sequences. And then if we can have a little bit light off, that would be great. And then, perfect, thank you so much. And then here you can, for example, genotype a patient on the SNP that you are interested in. If the patient is AA homozygote, you will see just T, just red signal. If a patient is GG homozygote, then you will see green signal. And if this is a mixture, a heterozygote, then you will see yellow. And I'm sure you can see green, yellow, and red over here. So this is a microarray that can be used for various purposes. First of all, with microarrays, you can do expression analysis. Right now, you can do classically about 40,000 transcripts in a single experiment. On the chips that we use, we can load 12 patients, and we can run 4 microarrays at the same time. So we can do about 50 patients per one run. If it's organized well, you can do about 150 patients, and you can get 40,000 transcript information in these patients, quite a lot of information. If you go for another technology like Affymetrix, they invest a lot in microarrays, so you can even do exon studies, not exon, but exon studies, and then you can cover about 500,000 of transcripts. So really a lot of information that you can get from this kind of experiments. If you do genotyping, right now it's possible to genotype up to 4 million polymorphisms, again, in 150 people per week. Some custom-made microarrays are available. Coding region-centered microarrays are available. So this is an affordable option in comparison to next-generation sequencing experiments. Not only you can do expression, not only you can do genotyping, but you can also do methylation analysis. So you can touch upon epigenetics, and you can get 850,000 methylation sites in a single experiment, again, 150 people per week. With cytogenetic arrays, you can look for large insertions and deletions, and there are arrays that can do 850,000 whole genome sites per one experiment. However, many people have told us that microarrays are dead. Microarrays are dead because whatever you can do with microarrays, you can do with next-generation sequencing, and you can do it better. Microarrays have known issues with reproducibility, with accuracy, with sensitivity, and they are very good tools, but there are better ones available. The important problem with microarrays is that they can only show you what you ask for. If you don't give a probe on microarray, you will never get an answer. So you will not find new SNPs, for example, new polymorphisms, no new methylation sites, no new cytogenetic insertions and deletions. And nowadays, the cost of doing a microarray experiment is not that much lower than doing a next-generation sequencing experiment. Very briefly, what is next-generation sequencing technology about? You start with your DNA molecules floating in the solution. You add the adapters, and these are universal adapters. So then you can do a PCR amplification reaction, and you can get much more of the same DNA without even knowing what is inside. That's important. You don't have to know upfront what is inside. The reaction that comes next is called bridge amplification. An important thing that you should remember is that now, with the SMART technology, we move from floating DNA molecules in the solution to the molecules that are attached to the surface. And once they are attached to the surface, you can clonally amplify them, and then you can add fluorescently dyed nucleotides, use a laser to infer the sequence of the molecules at given spots. So basically, microarray scanner is a kind of a high and fancy microscope or camera, if you will. You can repeat the steps many times, hundreds of times, and you get the sequence of the molecules that were previously unknown. You use bioinformatics to actually align these sequences along the reference genome, like the known sequence of a human genome or mouse genome or rat genome. And I'm not sure you can see it here. In the reference genome, there is a letter called G. In our sequenced molecules, we see also one G, but we also see A's. So this means that there is a polymorphism in this spot, and this person is a heterozygote. Okay, here we go. It's a short comparison of a sequencing capacity that is now available. The one that we have at Omicron is the smallest sequencer. It can still give you 15 gigabases of sequence. These are not gigabytes, these are gigabases. So you can get 15 billion nucleotides from a single run of this sequencer. And on the other end, there are high-tech X10 machines, real behemoths, which cost $10 million each piece, and which can sequence 18,000 whole genomes per year. 18,000 whole genomes. So they were advertised by Illumina as sequencing for the nations, sequencing for whole populations, even for the nations. Just to be fair and show the comparison of other technology, which with their biggest instrument is still a bit smaller than what we can achieve with the smallest instrument on Illumina side. However, it's worth mentioning that this other technology developed by Ion Torrent by Life Technology is also a very smart one. It's just a very expensive pH meter, which costs about $150,000 or so. When you add nucleotides, protons are released, and these machines are accurate enough to measure the current elicited by the flow of these ions. So if you know which base you are adding, you will know what is the sequence that comes out of this sequencer. I will tell a few words about other methodologies that are already available. This is PacBio, Pacific Biosciences sequencer, that is using something which the company called SMART sequencing. SMART sequencing standing for single molecule real-time sequencing. The two major advantages of this technology are that you can do it without PCR, that's first, and then you can do it in real time. So it's very fast, and you can get very long molecules. 15,000 base pairs, 20,000 base pairs. The disadvantage of this machine is that it weighs about 1,000 kilograms, so you better put it in the basement, otherwise if there is a crowd of scientists coming to see an interesting experiment, you may end down in the basement. Anyway, a new technology that is coming, developed by Oxford Nanopore Technologies, which this company developed a way to measure the current going through something which is called nanopores. And if you see if there is an obstacle inside the nanopore, there will be a smaller current going on. The bigger the molecule, the smaller the current. And you can believe it or not, but this machine is good enough to discriminate adenine from cytosine, from guanosine, from thymine. So you can just thread the DNA molecule across the nanopore and get the sequence of that. This is how it looks like. It's hard to believe these chips are not available fully commercially, but they were released and many scientists had a chance to do real experiments on that. And that's clearly the future of genomics. That's a disposable sequencer about the size of a pen drive. It's not a mock-up photo, it's a real photo. This is a sequencer. You add blood here, you get a sequence pretty soon. Okay, final technology I would like to tell a few words about is mass spectrometry that we use in our laboratory for proteomics. Mass spectrometry is a kind of a very fancy scale that can weigh molecules, can tell you what is the ratio between the weight of a molecule and the charge of this molecule. So something which we call M/Z ratio. The machine looks like that. It's coupled with HPLC, high performance liquid chromatography, to split the molecule into many single peaks of proteins. And then you inject it here, it flies through, and if you measure the time of flight, you can infer a lot of information. what can you get from this information? You can do quantitative analytical experiments, and as you can see, we are getting, in many experiments, we are getting close to 1,500 proteins measured quantitatively in a single experiment. What can it also do? It can fragment larger molecules into smaller molecules by colliding them with helium gas, and molecules break. And then if they break, you can retain them in something which is called an ion trap. You retain them in ion trap, and then you can still sequence it, so to say, sequence it again and again. You can do it many times. And it's very good for measuring protein modifications. So you can do something like glycation modification or phosphorylating modifications. We do something like a phosphoproteome. So all molecules that have been modified by phosphorylation of a protein molecule. Okay, so this is part one, technology, very briefly. Now I would like to tell you about a few applications, about experiments that we are doing right now in the Omicron laboratory. I cannot be too specific because these are ongoing projects, but just to give you a flavor and whet your appetite, what can be done in current biomedical science. So for example, an observational study in peripheral leukocytes in patients resistant to therapy. We're doing about 160 patients, 40,000 transcripts per each patient, and analyzing what causes resistance to therapy in this group of people. Another project, genotyping project, we have a custom selected polymorphism, about 400 polymorphisms in one of the autoimmune diseases selected to test if there is an association between any of these polymorphisms and the disease. And again, this is again a project that takes more than 1,000 samples. We hopefully want to accomplish that in 3 months. At least from our calculation, it's doable. How we will do that, we will see. Another project that we run in diabetes is to do 200,000 polymorphisms for each patient. 200,000 polymorphisms, but then we use a technology or bioinformatic technology called imputation. So we can really expand from 200,000 SNPs to millions of SNPs in this simple kind of experiment. Other experiments that we do, cytogenetic experiments in tumor specimens, comparing normal and tumor tissue. And also what is very important and very promising is joint analysis of proteins, of cytogenetic changes, of expression changes, of methylation patterns. Can give you a lot more information than if you just look at only one of these modalities. In the field of next generation sequencing, I told you microarrays are dead. So you know, these are experiments that still look interesting, but the future is in next generation sequencing. So we do, for example, a small gene panel of about 30 genes to discover new mutations in a monogenic disease. And we do this in an international collaboration with about 1,000 samples going on. Another project we use it is to sequence a large panel of genes, about 5,000 genes per patient, 5,000 genes per patient, to see the syndromes, to try to discover the nature of neurological syndromes that cannot be easily found just by looking at the patient, just like examination, physical examination of the patient. NGS is not only for DNA, it's also good enough for RNA. We do microRNA sequencing for matched samples of tumor and healthy mucosa adjacent to the tumor. We also search for fusion genes, which are important propelling development of various cancers. Very promising projects in metagenomics when you sequence bacterial samples, and this was initially something that we didn't even think about. Some people came to us, we started sequencing, went very well, and now we have more than 100 samples, metagenomic samples, showing the differences, for example, between various groups of patients. Amplicon sequencing, if you have a well-developed well-developed hypothesis and you just want to sequence one patient. Also targeted resequencing of the genes previously implicated in cancer development. And proteomics, our proteomic people always tell us, forget about this. Genomics is not important. Proteins is what they really do the job, and we should always think about proteins. So we try to think about proteins, and as I told you before, we can now identify about 1,500 proteins in each sample that we analyzed, for example, of cancerous tissue. And we also use ProteoMiner. This is a technique that can enrich your sample in proteins of rare abundance. It's very important because if you look at the plasma, serum, or any other tissue, you will quickly notice that there is a bunch, maybe a dozen of proteins that will make up about 90, 95, 97% of proteins in your sample. Like if you take blood, if you take plasma, you will have albumin, globulins, transferrin, ferritin, and then when you name about a dozen of them, then you have seen about 90% of the protein content. However, what seems to be the most important is these proteins which are very rare, and we have to enrich for them, or in other words, deplete the most abundant proteins, and this can be achieved with ProteoMiner, the technique that we used. Okay, so this was the application part, and now I want to tell you about the road ahead. So as you can see, our Omicron vehicle got some dust. Maybe some scratches on the surface. That definitely the door is open for various kinds of collaboration. Everybody is invited to hop on and join our band. And I still think the Monument Valley is ahead of us. Okay, so I don't have a crystal ball. I can't tell you what will happen in genomics in the next few years, but I can tell you what I would like to see. Because we can ask ourselves a question: is genomics mature field? Mature, like, for example, field of monoclonal antibodies, field of PCR applications, even the field of microarrays. Definitely we will see some improvements in PCR, in monoclonal antibodies, but nobody is really expecting major breakthrough coming out of this field because they have matured and they deliver, but the treasures are not there, I think. So in my opinion, genomics is not a mature field. What I would like to see definitely is point-of-care genomics, something that you can do on the spot with your patient. We have, for example, a project in septic patients. So as you probably know, 70% or more of bacteria cannot be cultured. You cannot get them in culture. If you ask the patient who is febrile and you ask what's causing this, in most cases you will not get an answer. You will probably culture something, but that will be like skin bacteria, not really the causative bacteria. So something that you can use for fast diagnosis would be fascinating. Single-cell genomics. That's happening right now, that you can split your sample down to a single cell. And we know that, for example, tumors are so heterogeneous that you have various clones of cells within the tumor. And you don't know which clone will prevail, which will be the most important 3 months from now. If you knew that, you could take some preventive measures to take care of that. Liquid biopsy. You can do by liquid biopsy, we simply mean measuring nucleic acids and proteins in repeated samples from blood. And it is well known now that, for example, cancer tissue releases its DNA into the bloodstream. It's in tiny amounts. But if we have sensitive enough instruments, we can probably get the diagnosis much sooner. And just imagine that you can sequence all that you have in your urine, stool, blood every week, and then you can see your cancer coming 2 years earlier before you see any symptoms in the clinic. Disease avatars, very interesting issues. For example, immune-compromised mice, which you can transplant with cancer. You can make many of these mice and then treat these mice with various drugs and choose the one that the patient will best respond to. That's something fascinating because you are not doing any harm with chemotherapy, you can decide which chemotherapy to choose. So one option is these mice, which are transplanted with tumors. The other option is organoids, very, very smart development in the past several years to be able to grow three-dimensional, for example, intestinal organoids in the culture. These three-dimensional cultures contain all cells of a normal intestine. It's not like just having colonic epithelium without having Paneth cells and other types of cells in the same culture. Here it's possible, and again, you can, for example, test for drugs that a patient will respond to before you give these drugs to your patient. This wouldn't mean much without genome editing technologies. These technologies are now available. CRISPR-Cas9, the name you should be familiar with, technology that allows editing the genome at the point where you want. So if you have a person with a mutation, you diagnose this person has a mutation, if you revert this person will be healthy. Of course, again, this is a very immature technology yet, because you cannot deliver this to humans. You can deliver this in cell culture, it's quite successful. You can do some experiments in animals, but it's not deliverable in humans. Also, we need a truly clinical sequencer, something that we can start anytime we want. Right now, we have to accumulate samples. If our run contains, let's say, 48 samples, we have to wait until we have 48 samples. Otherwise, we can do experiment earlier, but that will cost much more because we will be using a kit for 48 samples just with 20 samples. And in the long term, you cannot afford that. So we need a truly clinical sequencer. And this is homework for you, actually. What is on your list? What would you like to see on the list to genomics to flourish? And to me, a landscape of genomics is not mature. It's a wild, beautiful landscape. And if you look carefully enough, if the light would go down for a moment, you can see roads here. You can see roads here, and if the light would go even further, I think you can see a car traveling on this road. I have seen it on my computer, so I hope you can agree with me that there must be ways to get where we want in genomics in the few years to come. And I think I have managed. It's quite amazing. And I really thought that I will have to cheat on you and do a 1-hour lecture, but I did it in 20 minutes. So thank you very much, and enjoy the rest of the conference. The best is coming.
And now I would like to ask Dr. Jerzy Friediger, Professor Tomasz Guzik, Dr. Bartłomiej Guzik, to announce the winners of the special prize named after Professor Teresa Adamek-Guzik.
Thank you very much for coming here today also. For several years now, after Professor Teresa Adamek-Guzik passed away, we each year admitted one paper which was submitted to this conference, a special prize. The special prize which was taken from internal medicine subjects, simply because Professor Adamek-Guzik always took a huge care into this particular subjects. To this year, this prize, a special prize, because this is also a financial prize, and this is this year from last year also given by the medical chamber, regional medical chamber that we work also in, to student scientific circle with gastroenterology and hematology clinic in with the gluten and lactose and silent enemies and patients, to all the Kaczmarczyk, Justyna Gwóźdź, Patrycja Kutynicka, and Mateusz Fiema. Please come in. Thank you very much. And this year also, the Regional Medical Chamber admitted a special prize, which was not giving yes to any other paper submitted. This is a special prize because this prize is thanks to that you are focusing also not only on research, which is extremely important and you are doing it perfectly, but also on history and on a master's that used to work and used to teach us. And the paper was Andrzej Bobrzyński, first laparoscopic surgeon in Krakow, by Janusz Dragan and Maciej Nowak. If you're here, you didn't know that for the prize because it was decided this afternoon. We'll contact them and really hope for you also to, within your research, to focus not only on particles, but also on history and our people who are teaching us and are teaching you, and really, really, it is extremely important. Congratulations to them. We'll contact them and we'll give them the prize and hope now you'll get all the nice prizes that the organizing committee is awaiting for you.
I would like to ask Professor Tomasz Grodzicki and Professor Tomasz Guzik to award all winners of this year's conference. So we would like to award coordinators and winners of anesthesiology, intensive care, and emergency medicine. Yoon Hee Lee, Jakub Frączek, Magdalena Duchińska, and the first prize, Michał Krawczyk, Piotr Pałaczyński, Anna Smelik, Michał Długaszek, Szymon Haza. Second prize, Sylwia Batyzel, Katarzyna Konarska, Grzegorz Biedroń, Marta Sałek, Aleksandra Surowiec. And the third prize, Andrzej Hałek and Kamil Polo. And now I would like to award all coordinators and winners of Basic Science session. Maria Koshi, Kinga Michalewska, and the first prize, František Sanak. The second prize, Daniel Adams. The third prize, Aleksandra Dudzik, Kamil Grabowski. The first distinction, Martin Bortlik. The second distinction, Michał Długaszek, Anna Krywut, Ewa Olszańska, Justyna Musiał, Jowita Adamczyk, Paweł Ziora. Cardiology session. Coordinators, Marta Olszewska and Grzegorz Kiełbasa. For the first prize, Anna Różnowska, Anna Maria Nowacka, Anna Maria Przystup. Second prize, Grzegorz Kiełbasa, Katarzyna Bobucka, Alina Cempa, Marta Olszewska, Przemysław Pękala, Maciej Byrczak. And ex aequo, second prize, Anna Średniawa, Marek Stopa, Katarzyna Żabicka, Agnieszka Woś, Bernard Bukowski, Magdalena Ulman. First distinction, Wacław Morson, Dominik Spałek, Bartosz Mroczyk. Second distinction, Aleksandra Buczyńska, Magdalena Godlewska, Karol Kasprzycki, Aleksandra Pizun, Marcin Sułkowski, Marta Tomica. Cardiosurgery and invasive cardiology session. Coordinator Krzysztof Sojecki. First prize, Katarzyna Piątek, Mateusz Hołda, Mateusz Kozie i Jakub Hołda. Second prize, Jakub Konieczyński, Jakub Frydrych, Katarzyna Moczała, Marta Wrońska. The third prize, Aleksandra Gąsecka. Case report session. Coordinators, Paulina Frączek, Martyna Gomulska, Iwona Pena. And the first prize, Elsie Chan. Second prize, Agnieszka Gorycka, Sylwia Niewiara. And the third prize, Anastazja Bizjukowa, Indra Lipska, Anna Gutmann. And the first distinction, Artur Kośnik. Second distinction, Monika Emilia Król, Anna Malinowska. Dentistry session. Coordinators, Monika Szczepan, Omar Al-Obaidi. First prize, Aleksandra Sasuła, Anna Murzyn, Daniel Dobras, Katarzyna Kowalczyk, Marta Kilanc. Second prize, Magdalena Świątkowska, Joanna Kargol, Aleksandra Kołodziejek. Third prize, Magda Lipka, Katarzyna Łuczak, Joanna Uniewska, Ada Braksator, Anahit Chowanisyan, Marysia Białczak. The first distinction, Joanna Uniewska, Anahit Chowanisyan, Anna Strużyńska, Wiktoria Stojak. Second distinction, Joanna Uniewska, Anahit Chowanisyan. Dermatology session. Coordinators, Katarzyna Tyrak, Mateusz Mleczko. The first prize, Kamila Migacz, Katarzyna Tyrak. The second prize, Bartosz Balcewicz. The third prize, Mateusz Mleczko. And forensic medicine session. Coordinators, Małgorzata Kozioł, Karolina Strojny. The first prize, Grzegorz Kiełbasa, Przemysław Pękala, Katarzyna Bogucka, Alina Cepa, Marta Olszewska, Anna Kędziora. The second prize, Marta Krzywoń, Magdalena Stec-Polak, Karolina Polak, Rafał Świstek. The third prize, Maciej Dorotniak, Katarzyna Jasińska, Nadia Sajuk, Grzegorz Słota. The first distinction, Katarzyna Pejka, Karolina Strojny, Małgorzata Kozioł, Ewelina Szybiak, Karolina Lasoń. The second distinction, Krzysztof Kurek. Gastroenterology session. Coordinators, Karolina Jaworska, Mark Simpson. The first prize, Olga Kaczmarczyk, Justyna Gwóźdź, Patrycja Krynicka, Mateusz Szwiema. Second prize, Ewa Pękala, Kamil Wdowiak, Łukasz Juszczyk. The third prize, Martyna Gomulska, Paulina Frączek, Szymon Ciuk. The first distinction, Karolina Jaworska, Magdalena Małek, Dawid Storman. Second distinction, Noemi Cynthia Kis. Gynecology and obstetrics session. Coordinator, Aleksandra Dudzik. The first prize, Roddikina Valeria, Drobnice Anna. Second prize, Natalia Bajorek, Miłosz Sariusz-Romiszewski, Matthew Olesiak, Wojciech Rogula, Dominika Korzeniowska. The third prize, Olga Krzeczek, Anna Tomaszkiewicz. The first distinction, Ramona Galsone, Ilya Dmitrievs, Vitaly Skorsakovs, Evia Strankale. Second distinction, Kevin Fernando Venegas Hidalgo. Health science session. Coordinators, Iga Wąsowska, Magdalena Baran. First prize, Katarzyna Pietkun, Maciej Nowacki, Joanna Simińska, Joanna Stocka, Karol Ogórkowski. Second prize, Iga Wąsowska. The third prize, Marcin Jakiel, Radosław Łupkowski. The first distinction, Yevgeny Kostiuszenko, Jessica Zafra, Mikhailo Medvedev. The second distinction, Anna Średniawia, Dorota Łucja-Jarczewska, Katarzyna Żablicka, Magdalena Ullman. Hematology and oncology session. Coordinators, Anna Markiewicz, Łukasz Skoczylas. The first prize, Jakub Miazga, Joanna Nestorowicz, Katarzyna Kolary, Agnieszka Pomykała. The second prize, Paweł Bryniarski, Natalia Lorenz, Michael Kozłowski. The third prize. Michał Dworak, Dominika Pawlus, Kamila Gołdyn, Małgorzata Kozikowska, Bartosz Wojciechowski, Mateusz Jankowski. The first distinction. Jakub Miazga, Katarzyna Kolary, Agnieszka Pomykała, Joanna Nestorowicz. Internal medicine session. Coordinators. Paulina Godzisz, Weronika Kwiatkowski. The first prize. Agnieszka Waksman, Mikołaj Maga, Maciej Janik, Olga Chrząstek, Mateusz Koziej. The second prize. Agnieszka Waksman, Tomasz Rynkiewicz, Mikołaj Maga, Magdalena Przybylak, Aleksandra Śliwińska, Aleksandra Dziewulska. The third prize. Michael Kozłowski, Natalia Lorenz, Paweł Bryniarski. The first distinction. Kamil Polok, Andrzej Hałek, Agnieszka Wolny, Karolina Borowska, Magdalena Duchińska, Jakub Frączek. Another distinction. Alina Cempa, Katarzyna Bogucka, Agnieszka Oszywa, Natalia Jonach. It is another distinction from the jury. Ewelina Szybiak, Mikołaj Maga. Another distinction from the jury. Mikołaj Maga, Agnieszka Waksman. And another distinction from the jury. Sylwia Baptyzer, Katarzyna Konarska, Grzegorz Biedroń, Marta Sałek, Aleksandra Surowiec. Medical law session. Coordinators. Daria Zwinczewska, Tomasz Nowak. And the first prize, Izabela Jałowiecka, Monika Storman, Jolanta Majchrzak-Król, Anna Tarczyńska. And now I would like to ask Professor Beata Tobiasz-Adamczyk to award the winners. Neurology and Neurosurgery session. Coordinator, Radosław Łupkowski, Maksimson. The first prize, Anna Kot, Aneta Kosowska. The second prize, Paweł Jarski, Wilhelm Masarczyk, Izabela Jakutowicz, Aleksandra Ignatowicz, Martyna Ostry, Paulina Kałuża. The third prize, Aleksandra Śliwińska, Dorota Nielaba, Aleksandra Wojnarska, Katarzyna Homa, Wojciech Chmielak. The first distinction, Zofia Ślosarek, Kamila Gołdyn, Martyna Ostry, Michał Dworak, Paulina Kałuża, Dominika Pawlus. The second distinction, Katarzyna Kaczmarek, Monika Słowińska, Aleksandra Kordala, Emilia Danel. Orthopedic session. Coordinators: Tomasz Rynkiewicz, Natalie Halvorten. The first prize, Agnieszka Gołąb. The first prize, ex aequo, Marta Romanowska, Maria Bugajska, Anna Bajek, Dorota Porowińska, Tomasz Drewa. And once again, the first prize, ex aequo, Małgorzata Kozioł, Helena Zwinczewska. And the first distinction, Bartosz Maciąg. Pediatric session. Coordinators: Joanna Hubert, Miles Barre. The first prize, Michał Rosa, Aleksandra Cora, Iga Leszczyńska, Aleksandra Pękacka, Patrycja Pieczka, Maciej Byrczak. The second prize, Katarzyna Florkowska, Martyna Kowalczyk, Mateusz Moścież, Dorota Napieracz. The third prize, Mateusz Mleczko, Agnieszka Bur. The first distinction, Marta Krzywoń, Paulina Frączek, Michał Szczepanek, Mateusz Hołda, Paulina Marszałek, Mateusz Wilk. The first in this session, jury decided to give additional awards. So we would like to give honor awards from the jury to Anna Gutmane, Indra Lipińska, Anastazja Wizjukowa. Second prize from the jury, Katarzyna Roguska, Dawid Piotrowski. The first distinction from the jury, Agnieszka Gorycka, Anna Tomaszkiewicz, Mateusz Radom, Aleksandra Winiarska, Michalina Mazur. And the pharmacy session. Coordinators: Mateusz Szczybyło, Patrycja Hałaszuk, Klaudia Sadowska. And the first prize. Anna Furgała, Anna Ziora, Aleksandra Kacz, Joanna Misikiewicz, Boris Brus, Urban Kosak. The second prize. Katarzyna Mikołajczyk, Monika Szumańska, Katarzyna Dziewic, Alicja Kosałka, Magdalena Ciupka, Piotr Leśniak, Ewelina Zych. The third prize. Maciej Pawelec, Krzysztof Pardo, Bogumiła Kupcewicz. And the first distinction, Gabriela Kubisiak, Ewelina Stawarska, Dawid Synak, Adam Torpus. The second distinction, Kamil Piska, Karolina Uznańska. Psychiatry session. Coordinators: Anna Tereszko, Róża Ludwikowska. The first prize. Adrian Chrobak, Katarzyna Siuda-Krzywicka, Grzegorz Psiwek, Anna Tereszko. The second prize. Anna Tereszko, Adrian Chrobak, Szymon Jeziorko, Grzegorz Siwek, Katarzyna Siuda-Krzywicka. The third prize. Hussein Guray-Biser, Faith Sak. And the first distinction, Igor Salopek, Sara Vukasovic. Surgery session. Coordinators, Anna Stachura, Artur Majer, Justyna Zbaczyńska. The first prize. Brandon Henry, Joyetta Roy, Piravin Kumar Ramakrishnan, Tore Borgenvik. Second prize. Knut Gagnat, Zofia Wojciechowska, Paulina Wyszyńska, Justyna Zybaczyńska, Mateusz Tenerowicz. The second prize. Aleksandra Kowalczuk, Michał Wysocki, Anna Mydłowska, Paweł Lis, Sonia Osadnik, Kamila Migacz. The first distinction. Magdalena Godewska, Karol Kasprzycki, Piotr Małczak, Kamila Migacz, Justyna Sajdak, Kamila Damczyk. And the second distinction. Anna Mydłowska, Łukasz Bomba, Aleksandra Mikunda, Justyna Sajdak, Piotr Malczak, Wiktoria Parfeny. Two sessions were too small could be opened, and for this reason, but coordinators were really involved in their organization, and for this reason, we would like to award them. So, coordinator of the radiology session, Natalia Lorenz. Coordinators of otolaryngology session, Marcin Sołtysik and Przemysław Pękala. Coordinators of the workshops. Doctor-patient communication workshop coordinator Paulina Godzisz. Coordinator of the USG workshop Jakub Frączek. Coordinator of the More Than Morphine Psychological Needs of Terminally Ill workshops Michael Kozłowski. Coordinator of the Judge a Product by its Label, Understanding Nutrition Labels workshop, Agata Suszyńska. Coordinator of the Cardiac Surgery workshops, Krzysztof Sojecki. Coordinator of the Tournament of Minds, Michał Błasz. Coordinator of the Tournament of Minds, Kamil Polok. Coordinator of the Dietary Supplements to Take or Not to Take workshops, Małgorzata Maraj. Congenital heart defects from diagnosis to correction workshop, Julita Zacharczuk. When food becomes an obsession, contemporary eating disorders workshop, Aleksandra Zając. Present Endodontic Current Trends and Perspectives Workshop, Przemysław Kustra. Hypnosis History, Patrycja Jenda. Neurosurgical Workshops, Kornelia Kliś. And now I would like to thank members of the organizing committee of this conference. Mateusz Hołda, Natalia Dudek, Mateusz Koziej, Dawid Bugara, Paulina Dusińska, Matthew Wiciński, Piotr Puszmierz, Kinga Michalewska, Anna Mydłowska, Katarzyna Paczkowska, Magdalena Przybylak, Monika Szczepan, Björn Wypke, Helena Zwinczewska,
And on behalf of the whole organizing committee, we would like to say thank you to our president, Katarzyna Konarska.
So I hope you enjoyed the conference during the last days and that we will meet in the April here in the next year. I announce that the 23rd International Medical Students Conference in Kraków is closed. And for one second, we would like to invite all of you to pictures. Come here.